So we're going to be covering topic 2. Where is this? So I'll be covering everything from like 2.1 to 2.7, and then I'll be covering 2.8 and 2.9 tomorrow with topic 8, because 2.8, 2.9 are like the SL prerequisites to understanding topic 8 metabolism. So we'll be starting that. So the first thing we're going to start talking about are the what molecular biology is and the types of macromolecules. So molecular biology, really, we're trying to understand biochemical and macromolecular pathways in organisms, and how like things like biosynthesis work, or different enzymatic functions, different pathways. So you have like 4 main classes of macromolecules. You have carbohydrates, lipids, nucleic acids, and proteins. Carbs are really important for cell communication and structure. They're like components of the cell membrane, and you can see them as like extensions on the cell as glycoproteins, which are important for cell recognition, junctions. Carbohydrates are also part of nucleotides. They're because you require pentose sugar for either DNA or RNA, which is ribose or deoxyribose. And energy. They're the most immediate form of energy use. Carbohydrates are going to be the thing that your body uses like before all the other macromolecule types. So, and then you have lipids. Lipids are also something you can find on the cell. Glycolipids are also on the cell, which are also used for communication and structure. Steroids are a type of lipids, so all those different hormones, different fats, different oils, different plant oils like olive oil. Oils are really associated with like animals, and fats are really oils are really associated with plants. My bad. Fats are really more associated with animals. Your phospholipids and cholesterol. Cholesterol is a major component of the cell membrane. I do believe you have to know how cholesterol increases the structure and the rigidity of the cell membrane. And then nucleic acids. The ones you have to know are about DNA and RNA. And then proteins. They're really important for transduction pathways, which are how do I say this? They're like cell-to-cell communication pathways, and then catalysis. So the components of each of these major macromolecule groups are going to be monosaccharides, lipids, proteins, and nucleic acids. Lipids are kind of weird because all of these other things are the monomers, but there's like no monomeric like constituent in the same way that like monosaccharides make polysaccharides, or proteins make polypeptides, or nucleic acid make like nucleic acid chains. So you have to know what monosaccharides are, disaccharides are, and polysaccharides. Did not mean to do that. And you have to know the different bonds that each are associated with each. So glycosidic linkages are associated with sugars. And then lipids, they have like ester linkages. There's different types of lipids. They're kind of like all over the place in terms of how complicated they are, but you do have to know about triglycerides and phospholipids. And then proteins, you have to know the general structure of a protein. I do believe you have to be able to draw the 2 different forms of glucose, and you have to be able to draw the general form of a protein. It's just the amine group here. Here's the carboxyl group, so this is the COH right here. Here's the variable group, so the variable group is going to be the thing differentiating the amino acids from one another. And then you have the alpha carbon right here. This is the main carbon that everything is attached to. And then amino acids link together via peptide bonds. Nucleic acids over here. Nucleobases are a form of nucleic acids. You have your pentose sugar. Well, nucleobases are a part of nucleic acids. I'm sorry. You have your pentose sugar. You have your phosphate. You have your nitrogenous base, which depends on which type of nucleic acid you're going to be looking at. For RNA, it's uracil. You have U, A, G, and C. And then for DNA, it's T, A, G, C. And those are held together by phosphodiester linkages. It's not just one bond, but I'll show you a picture, I believe, later of that. So metabolism is an important part of how an organism functions. So metabolism is the net enzymatic reactions of both anabolism and catabolism. So when I was learning chemistry and biology in high school, my teachers told me, remember catabolism as like, I don't know, cannibalization. So like catabolism is molecules being broken down, complex molecules being broken down into simpler molecules, and anabolism is you're building more complex molecules with simpler building blocks. So you have condensation reactions that are associated with anabolism. So those are going to be your dehydration synthesis. So you link two molecules together, and then you link them together by getting rid of one hydrogen on here on one side and then a hydroxyl group on another, and then that releases water. Hydrolysis is the opposite. That's associated with catabolism. You introduce water to a source to break the bonds, and here you can see that dehydration synthesis and hydrolysis are the opposite of each other in terms of reactions. You have to know about vitalism. Vitalism is like vitalism. So urea is like an organic compound, but it's something that can be artificially made. So vitalism is like organic molecules can only be made from living systems. Otherwise, we wouldn't have organic chemists. You get your urea by I think the synthesis is heating inorganic ammonium cyanate. But yeah, you have to know about Wöhler synthesis, and you just have to know that the fact that you can artificially make urea disproves vitalism. It's not a huge part of the syllabus, but it is like a little part of the syllabus. Okay, so I was talking about the drawings earlier. So for sugars, you have to be able to draw these two isomers of glucose. You have to draw alpha D glucose and beta D glucose. The only difference is like this, the alpha carbon here. So you have to, they're basically the same thing, but the OH and the H group are just flipped on these. You have to be able to draw that in the ring form. And then I did talk about the drawing of the amino acid. So alpha carbon here. You have to get the sides right, by the way. So your amine group has to be to the left, and your carboxyl group has to be on the right. Your variable group, and don't forget this little hydrogen down here. You don't have to memorize like the different R groups. You just have to like write R for variable group. There's way too many R groups. You only need to know R groups if you're doing really, I guess, college level biochemistry. On top of the different isomers of glucose, you do have to know this one form of ribose. It's D-ribose. D-ribose is also like similar to these, except has this OH group here. It was very similar to beta glucose in terms of structure, but yeah. I just practiced drawing these for like, I don't know, like a while before, just like muscle memory to help me remember them for the exams, because they'll just ask you to draw these on the exam. This is also like the general form of what a lipid will look like. It's just really long hydrocarbon chains, H at the end, and this carboxyl group at the beginning. So now we're going to talk about water. Water, there's a lot of like things you have to know about water. So I'll talk about the really basic chemical properties of water so you can understand how water is able to do what it does. So the reason why water is so effective in all of the things it really does is because it has a electronegativity difference. So like this oxygen is more electronegative, because more electronegative than your hydrogens here. So it's going to be pulling the density of the electrons towards it. I think it's easier to remember if you think about how oxygen has a lot of protons associated with it in comparison to hydrogen. So the protons of the oxygen are going to be pulling electron density towards it. So oxygen is going to be a partially negative side here, the partially negative dipole, and then since the electron density is being pulled away from your hydrogens over to the oxygen, these are going to be your your partially positive dipoles. And you also have to know that hydrogen bonds are able to form when these hydrogens here can bond to nitrogen, oxygen, or fluorine. These hydrogen bonds are a type of intermolecular force, so it's not going to be like chemical bonding like this per se, but general associations. But yeah, the overall polarity is a very important thing about water. So I think the difference between hydrophobic and hydrophilic molecules are pretty simple to understand. So hydrophilic things they freely dissolve and dissociate in water, so you have to think about the thing of like dissolves in like, so polar molecules will dissolve in polar molecules, and also things like salts. This is what a salt looks like in water. The partially negative and partially positive things will associate with like the opposite charges. And the hydrophobic things don't really dissolve well in water because it's, remember, like dissolves in like. The reason why polar molecules are able to dissolve in water so well is like, so this is the partially positive dipole. So like the negatively charged ions here can associate with this, and then the positive ions can associate with this, and that pulls those ions away from bonding from each other, or like those polar molecules can associate with different parts of water. That's also why things that are nonpolar, like hydrocarbons and fatty acids, don't really dissolve that well in water. I'm sure all of you know that oil doesn't really mix well with water, it just kind of like forms layers. The reason why is just like it's let me show you. It's a bunch of nonpolar associations. That's mainly because C and H, there's not a really large electronegativity difference, so it's going to be not very polar. So it's not going to be, it doesn't want to be dissolved in water, so it's going to stay separated from water. So things that are nonpolar, a lot of proteins are nonpolar, but this is really dependent on the R group. Some of them can be polar R groups. R groups are what determine if it's going to be polar or nonpolar. Cholesterol, really important for it to be nonpolar, so it stays within the cell membrane, and phospholipids. So you do have to compare water to methane. I'm not exactly sure why methane, but that's the example the IB wants you to know. It is probably because they're comparable in a lot of things, like comparable size in terms of molecular weight, and then they have similar enough electron geometry, similar shape, and similar bond angles. Very minimal difference between their bond angles, but the main difference is the fact that this is polar, but this is nonpolar. And methane forms weaker dispersion forces, while water can form intermolecular bonding, and this is all down to polarity. If you look at the carbon, it's just going to be pulling electron density towards itself. It's going to be coming from this way, this way, this way, this way. It's all equal towards the carbon, but the poles are forming in different ways on the oxygen, if that makes sense. And it's because of the polarity and water's capability of forming polar bonds and hydrogen bonding that polar has much higher boiling point and melting point, because water is able to form these intermolecular associations with itself, and it's really hard to, you have to add energy to get the molecule to boil and melt, while methane does not want to have intermolecular associations with itself. So that's what gives water higher melting point, boiling point, and enthalpies of fusion and vaporization. That's also why water is really good as a coolant. That's why humans have evolved to have water as a coolant when we sweat, because it helps cools us down, because you have to add a lot of heat to water to heat it up. So we're kind of bouncing all over the place, but this is how like the IB syllabus, in terms of like the standards, organized it. It's a little weird, but I think we're going to be talking about the different macromolecules. So with carbohydrates, they're linked together through condensation reactions. This is not like the only thing. Condensation reactions are just the things I was talking about earlier. All of the macromolecule groups use condensation reactions or dehydration synthesis. You have to know you have to know like the different names for these processes. So condensation reaction and dehydration synthesis are the same. Sucrose, lactose, and maltose are the three disaccharides you have to know. And then you also have to know the different functions of the forms of sugars. So monosaccharides, glucose, galactose, fructose. Fructose is in literally everything. Like almost all the sweet things you have high fructose corn syrup. Monosaccharides are what your body requires. So like they'll break down disaccharides and polysaccharides into these simple sugars for us to be able to use them. Gives good flavor is because monosaccharides tend to be much sweeter and more palatable to our tastes than disaccharides or polysaccharides. Disaccharides are known as like a transport form, so that's lactose, sucrose, and maltose. And polysaccharides, they're more for like storage or like cellular components. Cellulose is part of cell walls. Glycogen is how we store glucose, but in animals. You also have to know this for like, you have to know that glucose is stored as glycogen in the liver in animals, but I think that's for a different unit. That might be human physiology. And then starch. Starch is also a major component of plants. That's how they store their glucose in the form of amylose and amylopectin and starch. It's a lot to like know, but it's a lot to know, but like it's I would say it's pretty doable if you just like make charts for yourself. So I just mentioned that cellulose is like a structural component, and then starch is like how they store glucose. Starch can be found. Starch is made up of different branches of like amylose and amylopectin. You have to know amylose and amylopectin. Amylose has these like it's like really branched. It forms like these branching structures. No wait, that's amylopectin, I'm sorry. Amylose forms these like helical chains, and then amylopectin's the really branched forms. These are both parts of starch, by the way. And then glycogen is how we store glucose in the liver. It's analogous to amylopectin in terms of sequence homology and structural homology. This is because they're both branched chains made up of alpha glucose, while amylose is made up of helical chains of alpha glucose. And then linear chains of beta glucose for cellulose. Remember those beta glucose and alpha D glucose and beta D glucose as the two forms of glucose that you have to be able to remember and draw. So like think about these concepts as you're like learning about them and drawing them. So we're moving on to fatty acids. So lipids are also a form of energy storage. They're more associated with long-term energy storage as opposed to carbs, which is like a more immediate form of energy use. So these saturated fats are usually found in animals. It's because saturated fats have no double bonds, so it's easier to stack these on top of each other. So like they're the same thing, so it's just easy to stack together because it forms really nice puzzle-like pieces that connect together. Trans fats are really usually not found in nature. Trans fats, they're unsaturated trans fats. Saturated fats can be trans or cis. Cis is on the same, so cis means same, and trans is on the opposite sides. So this causes like some branching here. These branches look the same, different. Since unsaturated cis fatty acids have their hydrogen molecules on the same side, it causes this bent formation, which is why unsaturated cis fatty acids tend to be better for you because they can't accumulate in the same way that saturated fatty acids can in the body. Most of like the cellular fat you see in animals is going to be saturated fatty acids. Unsaturated trans fatty acids are your more synthetic fats. If any of you have heard like health concerns over unsaturated trans fatty acids, this is the reason why. It's this right here. It's not as good as saturated fatty acids in terms of like storage, but if you put these on top of each other, they're able to store pretty well too. And then you have triglycerides. So you have to know about triglycerides. These are the largest in the different lipid families. They're associated with really long-term storage. So you remember I was just talking about how saturated fatty acids, they're able to like stack onto each other really well, and that's why they're able to be stored really well. It's like these long hydrocarbon chains. They're really stable, able to stack. You'd see that here, really long hydrocarbon chains, and they're really easily stackable. I don't think you really have to know the dehydration synthesis reaction for these. You might have to. I'll double check if you need to later. But triglycerides, you have this glycerol molecule, which is like a sugar alcohol. Then you have the main fatty acid, and then dehydration synthesis to make your triglyceride molecules. So I was just talking a little bit about the health risks associated with all these fats, especially heart disease. That's really prevalent. So saturated and trans fatty acids increase cholesterol. Remember, it's really easy to store these in the body, so they're going to accumulate in the body and increase the cholesterol content in your body, while cis fatty acids decrease them. So these fats and cholesterols have to be associated with lipoproteins in the blood. So low-density lipoproteins carry cholesterol from the liver and it circulates in the blood, while high-density lipoproteins bring excess fats to the liver for disposal. So you want high HDLs and you want low LDLs. So saturated fatty acids increase the LDLs, and so with trans fatty acids are especially bad because they also decrease the HDLs. If you've ever heard of like people having health concerns over trans fatty acids, this is like a big reason why. It not only increases the amount of LDLs in the body and therefore cholesterol, it also decreases the amount of HDLs. So it's less of those fats are going to be metabolized in the liver and brought out as waste. So it significantly increases your cholesterol. Cis, they're really good because they increase the HDL and therefore decreases the cholesterol. If you have high cholesterol, you can get this condition called atherosclerosis, which becomes chronic heart disease. Atherosclerosis has to do with like the arteries and the veins. Sorry, I was just like thinking. Atherosclerosis, you have these like buildups of fats, cholesterols, and like other things in the artery walls, and then that clogs the artery walls. That's how heart attacks happen because you have this buildup of plaque on the artery wall, and that restricts blood flow. So for the health claims and the evidence, foods associated with high saturated and fatty acids, trans fats, in studies they've been linked to increase outcomes with chronic heart disease, and the reciprocal is true in studies with where we have patients consuming a lot of cis fats. The prevalence for chronic heart disease is much lower than control groups or their contemporaries. Some populations are exception for this. This is mainly present in some of the hunter-gatherer tribes of Africa, or the hunter-gatherer peoples of Africa. There is some evidence that is against this. Lowered saturated and trans fats consumption, but you still have higher chronic disease risk in the past few decades. But this is really associated with high carb intake, and chronic heart disease is like a really intersectional issue because I mentioned earlier, fats aren't the only thing that can be associated with atherosclerosis and the buildup of plaque in your arteries. Other things can also build up in your arteries. You don't have to know a ton about this, but just like memorize the things that I just mentioned because I tried to like minimize how much you'd have to look into like the studies and stuff. And now for BMI. This is like a really simple thing. BMI, you usually just take like a ruler and then like from one side to another. So like if your weight is like 150 kg, you put this ruler on one side, and then your height would be like say 75, and then wherever the ruler hits in the middle over here is like your BMI. So different BMI classes. So from here to here is overweight. From 20 to over here is acceptable. So your BMI is mass in kilograms divided by height in meters squared. BMI is not exactly accurate in people with a ton of like muscle. It's pretty common for pro athletes that have to bulk up on weight to be considered obese or overweight by the BMI scale, which is why, like any metric, you can't use it in a vacuum, and other health outcomes are considered when we're looking at obesity and weight in patients. So next, moving on to amino acids. So there's 20 different R groups. You don't have to memorize any of them. I have to, which I'm salty about. So these different R groups or side chains, they give the amino acids their properties. So you have like polar side chains, non-polar side chains, acidic side chains, non-acidic side chains. You have some side chains that have sulfur, which is a really important property for biological molecules. These side chains will affect the acidity, the polar, non-polar, basic interactions that within itself and with other amino acids that will affect how a protein folds. So like proteins, because how do I explain this? So things that are polar and polar want to associate with themselves, so like they'll fold towards each other, and that's true with non-polar. So one of the things you do have to know is you have to be able to draw the dehydration synthesis or condensation reaction for transpeptidation. So you have to just get rid of one OH group here and one hydrogen, so water here. Just know this. It's easier if you practice drawing an amino acid and you remember the structure for an amino acid, and then you get to drawing dehydration synthesis. So different forms, you have different conformations. So primary is just like the amino acid sequence, like glycine, valine, just like in the line. And then secondary is the alpha helices and the beta pleated sheets. You have to know alpha helices and beta pleated sheets. And then tertiary structure, and then quaternary structure. Tertiary structure is when you start having a polypeptide. Quaternary structure is when different polypeptides associate with each other. So in terms of proteins, genes have the collective, which is the genome, and proteins have the collective, which is the genome. The proteome library tends to be larger than the genome library because there's different ways you can cut exons and introns through alternative splicing and different post-translational modifications. Post-translation modifications are really something you're going to be seeing in eukaryotes, same with alternative splicing. But I don't think there's any alternative splicing in bacteria, but there is some post-translational modifications in bacteria. So you have to know examples of proteins. So structure, collagen. Collagen is a part of your hair and a part of your hair. And spider silk, hormones, insulin, that affects your blood sugar levels. Also other hormones like estrogen, testosterone. Then you have immunity. So immunoglobulins, these are the BCRs or B cell receptors on the surface of B cells. So those are your different antibodies. You will have to know this for human physiology. Sensation, rhodopsin, and then you have actin and myosin. That's also something I think you have to know for human physiology. And then rubisco. Rubisco is an important part of the Calvin cycle. It's responsible for carbon fixation in the light independent cycle to produce sugar molecules. So you're taking a 3-carbon molecule and you're trying to turn it into a 6-carbon molecule. So yeah, you also have to know about rubisco in really specific detail. I think immunoglobulins, actin, myosin, rubisco, those have like associated things of themselves. This is for human physiology, and then this is for metabolism. Metabolism we're going to be covering next session. So for proteins, most enzymes are proteins. You could have special ribozymes, but most proteins are going to be the bulk of your enzymes. So enzymes function by lowering the enzymes are responsible for catalysis. So they're going to be responsible for catalyzing different reactions. So it's going to be based on collision theory. Collision theory is that things have to collide with the right orientation and the right energy for reactions to occur. Because different active sites are on different parts of the molecule. So if you like collide, but it doesn't collide with the right active site, the reaction can't occur. So you have to have the correct orientation. So the main factors you have to know are pH, temperature, and concentration, and how they affect enzyme activity. So remember, all enzymes have a optimal range, and once you start leaving the optimal range, the rate of reaction and therefore their effectiveness, their rate of which they can catalyze things, reduces. Their function is reduced. So pH is really just likes the sweet middle spot. Don't get too acidic, don't get too basic. Temperature, if it's too cold, there's not going to be enough energy for a enzyme to function. If it's too hot, also if it's too cold, there could be crystallization, and that can damage a protein. If it's too hot, you can have the bonds, the hydrogen bonds in a protein start to denature and it starts losing its shape. Right here is where you have the optimal performance. Note that the optimal performance slightly favors warmer temperatures. This is just because if you add extra heat to the enzyme, you're going to provide it with more energy, and that also more collisions are likely to happen because the molecules are colliding at a faster rate. And then substrate concentration is just this right here. So this is just determined by the more substrate you have, the more the rate of reaction increases until it drops off at a point. This is because there's too much substrate, and increasing the substrate at a certain level doesn't increase the rate of reaction because your enzymes are going to be all occupied. So industrial enzymes. This is like part of like the field I'm studying. I'm one of my majors is biotechnology, so like industrial enzymes are like my thing. So these are like different enzymes. So industrial enzymes, we really one of the applications for enzymes is immobilized enzymes. I do believe you have to know immobilized enzymes as like a major industrial application. So they're going to be attached to a surface. It's usually some type of column that's insoluble and inert. You want it to be insoluble because you don't want it to wash down. You want it to stay where it's at. And this is really good because we can keep the enzymes for later use. So we can reuse the column after we wash it. And then that allows for better separation of the substrate because it's going to be all immobilized. It's not going to be like flowing in liquid. This is like a huge thing because reusing is really big for us because it's really expensive to get a lot of enzymes and proteins. Some of the enzymes I've worked with are a few $100s, a few $1,000s per bottle. So yeah, reuse is a pretty big thing. There's many examples of immobilized enzymes, one of which is ELISA, which is like this really cool aminosorbent assay that detects for high concentration of HCG for pregnancy. You have to know ACG for pregnancy. I'm not exactly sure. You have to know ELISA for. I don't know if you have to know ELISA specifically for that part of human physiology, but you do have to know about HCG. We also have these cool things called biosensors that they're also immobilized, but they're biosensors that can detect different things in a patient sample. So like they're high sensitivity, specificity, pretty cost effective too because you can reuse them, and it's becoming a thing in like specialized and patient specific treatment plans. There's also other industrial uses for immobilized enzymes, such as biofuels, food production. I don't really know too much about biofuels and food production because that's like not a biotechnology thing, or well, it is, but it's not my part of medical biotechnology. The example you really have to know though is lactose-free milk. Mammals in general don't have lactase persistence, which just means sorry, mammals don't have lactase persistence, which means they just produce lactase into adulthood and maturity. That's why a lot of people are lactose intolerant as adults. They just stop producing the enzyme. So we can treat milk with immobilized lactase, and that lactase will break up the lactose, and then those people can drink lactose-free milk without having all the indigestion. Another cool thing is, I mentioned this earlier, but monosaccharides tend to be sweeter than disaccharides. So that milk tends to be sweeter. You have reduced crystallization in the milk because it's more soluble as a much smaller molecule. And it makes dairy production more efficient because you can reuse the enzymes. And bacteria tend to dairy production, like things in yogurts. It makes that more efficient because bacteria tend to ferment simpler sugars much better. They prefer to ferment simpler sugars, so you have better production of your yogurts and cheeses. Okay, so we're going to jump to the central dogma. Central dogma of biology describes the flow of information. So that's just DNA to RNA to proteins. So DNA and RNA are going to be the nucleotide polymers. They're pretty similar components. They all have this phosphate group, this pentose sugar, and then this nitrogenous base. The only real difference is different nitrogenous bases, and deoxyribose doesn't have an OH group here on the 2-carbon. You have to know this. It's a really important thing. This 2-carbon here, deoxyribose just has an H here, but for ribose it has an OH here. So they have pretty minor structural differences, but the fact that the OH here is actually a pretty big structural thing. The OH is prone to interacting and attacking other molecules. So ribose is more prone to being unstable and breaking down or interacting with other molecules. Deoxyribose is more inert because that H group doesn't lend itself to interacting with other molecules. So this is the reason why RNA just tends to be this transient form because in mRNA or the different other RNA forms like tRNA. And that's why DNA is where we store our genetic information. It's more inert, so it's much more stable. And also RNA tends to be single strand, like mRNA, tRNA. Well, DNA tends to be double stranded. You do have some viruses that are double-stranded RNA, but you don't really have double-stranded RNA in living organisms. Also, the nitrogenous bases are different. They have almost the exact same nitrogenous bases, but in ribose, in RNA, you have uracil, and then in DNA, instead of uracil, you have thymine. So DNA structure. So I just mentioned that the DNA structure is double-stranded. So here's one strand, here's another. This is the model that Watson and Crick built to elucidate the structure of DNA. Watson and Crick were the two scientists that used models to elucidate the structure of DNA. It's also anti-parallel, so one side is 5' to 3' and the other side is 3' to 5'. It's here. So the phosphate of one nucleotide makes a phosphodiester bond with the 3' hydroxyl, and then you have polymerization through multiple dehydration synthesis. So dehydration synthesis or condensation reactions. Like I mentioned, dehydration synthesis or condensation reactions are how you polymerize monomers into polymers. So this double twist, this helix here tends to be 10 to 15 base pairs per twist. And then Watson and Crick they had built their models on top of the discoveries of Phoebus Levine, Erwin Chargaff, and Rosalind Franklin. They're honestly pretty I thought they would be less controversial, but they're honestly pretty controversial figures. When I was taking molecular biology and quantitative biological methods in university, my professor mentions that like Francis Crick honestly got canceled because he's like really sexist, and I can see it because Watson and Crick are there's like a lot of controversy with the elucidation of the structure of DNA. You have to know Rosalind Franklin. Rosalind Franklin was this brilliant woman who elucidated the double helix nature of DNA. So she isolated DNA. She like made extremely pure samples, and then she fired X-ray beams at them. And then with those X-ray beams, she was able to see kind of like this X structure here. X-ray crystallography is like no joke. It is like one of the hardest things you could do. It takes at least a few years for you to be able to get the protein structure and crystallization to happen. And her work was like really passed on. Watson and Crick used her work without her knowledge, and when they won the Nobel Prize, it was those two. Franklin was not awarded the Nobel Prize, despite the fact that it wouldn't have been possible for them to build the structure here without her work, because she elucidated the fact that it was double helix, so it was like this kind of looked like this X shape under the X-ray crystallographer, and the approximate like sizes and distances between the base pairs, or like the length of the helices, like the minor and major groove of the helices. So you can see here also the base pairing that's happening between here, and then the sides here are really open, so they're not as close on these sides. So the sides are open here, and this picture doesn't show it, but you also have like protrusions of the major and minor groove. So those allow it to be accessible for replication, translation, transcription, and different DNA modifications to the bases that will regulate protein synthesis and RNA transcription. I didn't really talk about Phoebus, Levine, and Chargaff, but Chargaff is really important because Chargaff was the scientist that elucidated the fact that he was basically the person that discovered base pairing. So when he was doing his work, he discovered that there's an equal amount of A to C in an organism, or an equal amount of G to C in an organism. So he's responsible for base pairing, so base pairing here in the middle. And then Phoebus Levine was the one who overall elucidated the structure of protein of these nucleotides. So he was the one that discovered one phosphate group, one nitrogenous base, and one pentose sugar. So DNA replication. The process is semi-conservative. So here's the old DNA, and then you have like a replication fork that kind of opens up, and and then the replication fork opens up. The template strand is used to synthesize the new DNA strand. So you're going to have one old strand and one new strand, which is why it's called semi-conservative. You conserve one DNA molecule and you make another DNA strand. And this happens through base pairing. So if I open up one side, it's going to, and if it's like T matches with A, G matches with C. So that's how the nucleotides form. This semi-conservative model was elucidated by Meselson and Stahl. They used these radioactive nitrogen isotopes. They were trying to discover if it was conservative, where it's just all old DNA, or semi-conservative like this, or dispersive, where it's just like kind of random where the DNA goes. So this is what their experiment looks like here. So the light chains are the N14 carbon isotopes. You have to know this experiment in detail. The N14 carbon isotopes. So both strands are light, so it doesn't sink as much. And then both strands are heavy, so this is the N15 carbon nitrogen isotopes. If I've been saying carbon, sorry. This is the results they observed after 2 generations of DNA replication. They observed this, which is the mixture of light. No, sorry. This is like isolation, where it's just, this is what they observed actually. It's a mixture of light and heavy. So it's kind of in the middle from both. So this is how they discovered one half of it is old and one half of it is new, because the old would be like one nitrogen isotope, and then the old would be the other nitrogen isotope. So for replication, like I mentioned, you have this like replication fork that opens up. DNA helicase is going to be the enzyme that unwinds it and breaks the hydrogen bonds that are holding them together in the middle. So you form this like big fork here. So the leading strand is going to be the thing we synthesize from 5 to 3 prime. It's going to be here. And then the Okazaki fragment, it is going to be, it's much harder because this strand leads 5. This strand is 3 to 5, so you have to synthesize this way. And like synthesizing this way is much more awkward. That's why it's discontinuous. So you do have to know about the Okazaki fragments. So the main DNA, the main molecule that is going to be building this up is going to be DNA polymerase. So it uses energy from like these free dinucleotide triphosphates in the cell, and then it cleaves off 2 of those phosphates, which is a pyrophosphate. And by cleaving that off, that's going to be a release of energy, and then that energy is used to catalyze adding that nucleotide to the opposite side, according to base pairing rules. So an
Oh, wait, sorry,
application.
this is like a question. Can I ask one?
Yeah. Do you have a question?
Oh, yeah. So you spoke about Okazaki fragments. How long are they normally?
Oh, that's a good question. They tend to be like pretty short because it's a really weird thing for DNA. It does not like to do that. I'm trying to think. It's been so long since I've learned this.
Are they all the same size, or is there like a very differing length? And like, what's the average size?
It varies in organism to organism, or from different clade to different clade. In bacteriophage, Okazaki fragments in like bacteriophage T4 and bacteria are usually like 1 to 2,000 nucleotides long, which is pretty long. In eukaryotes, they tend to be like 100 to 300 nucleotides long, so it's much shorter in humans and like other eukaryotes. It's just really dependent on the species you're looking at, or like the genus you're looking at.
Wait, okay, so why are they different between eukaryotes and bacteria? Like, why does size matter that much?
Oh man, this is gonna be getting into bacterial genetics, and then I don't want to like overload you guys, but okay. There's a lot of things different in terms of I don't exactly know why actually. Uh, because like this is not a thing you have to know for IV biology. Um,
Okay, sorry, never mind then.
but it is an interesting question. I have never thought about it, but it's actually the actual reason I think is because we just literally don't know. We don't know why. There's like the field of like genetics, and like at least like modern genetics with like sequencing capabilities is like pretty new. We literally have no idea why the length is different. It's just a mystery to us. I think there's research going into it, but yeah, that's a pretty good question.
Right, thank you for that. That's understandable.
hmm Um, so an application for DNA replication is PCR. As a biotechnologist, PCR is like so cool. There's been many derivatives of PCR. You don't have to know this, but like I'm just speaking as someone who works in the field. This is like one of the coolest things I've ever worked with in my life. Like it's cooler than CRISPR. CRISPR's kind of a disappointment, in all honesty. But PCR is like one of the things that is so integral to our field right now. So PCR is called polymerase chain reaction. So it's because you have to run PCR for multiple cycles. We usually run it around 30-35 cycles of just like this process right here, 30 to 35 times. So you get like a billion DNA copies. The point of PCR is you're trying to take a fragment that you're interested in and you amplify like multiple like billions of times so you can like study it, use it in like experiments. So yeah, so the process involves like denaturation. So you like break the DNA strands apart and then you anneal like this primer to it. Primer design is like a whole thing in of itself that's really interesting. So you design a primer that can attach to like the DNA of interest that you care about. So the annealing part is really important because that means you can study like a specific DNA fragment that's of interest to you and not like study a bunch of like other DNA fragments. And then you have extension where you add molecules that can synthesize from those free nucleotide triphosphates. And then that's elongation or extension. It's the same thing. And then you repeat this like 30-ish times. You have to know about T. aquaticus. T. aquaticus is like this really cool thermophilic bacteria that lives in like hot springs. They discovered T. aquaticus living in like the hot springs of the Yellowstone, which is like hundreds of degrees hot. It's really impressive. It's Taq polymerase. We want Taq polymerase in specific because Taq polymerase can survive this denaturation process. Other polymerases wouldn't be able to survive it. In fact, Taq polymerase is like optimal at 70 to 75. Some of the higher end ones can be optimal at 80 degrees Celsius, which is like insane to me, considering like normal body temperature is 37 degrees Celsius. This is like the optimal range. So this is like a great molecule for PCR. And then we have transcription. So transcription, you're basically opening up those DNA strands here, and then you're making it accessible for RNA polymerase to come in and take this DNA and then make an RNA script. There's different forms of RNA. The only two main ones you have to know are like tRNA and mRNA, but like it's crazy to me that mRNA, it's not crazy to me. mRNA has like such a big focus in the syllabus when it's only 1 to 5% of all of these cellular RNA. There's like so many other types of RNA, like it's crazy. So
I just had a question about transcription. So, how exactly does the cell regulate where the polymerase starts translating the DNA?
So that's a good question. We usually have in humans we have like a sequence that can be recognized, and then that starts transcribing from there. It's really complicated in bacteria. Also, this is like, can you repeat your question? Sorry, I just lost my train of thought.
So basically, how does the RNA polymerase know where to start? Because if it starts at the wrong place, it's going to produce the wrong protein, right?
Mm-hmm.
So just curious about that, yeah.
So you always have like origins of replication. RNA polymerase has like something that can bring it over and that it can bind to. So like the chaperone can bind to like the specific place where it needs to start, which is the TSS, the transcription start sequence, and then it can start transcribing from there. If it doesn't It's like the promoter region. The promoter region is where RNA polymerase can recognize as a starting site, and those promoter regions tend to have sequences that consensus sequences. So yeah, promoter regions are the reason why. In bacteria, it's called the origin of it's not ORIC, it's operons, but you don't have to know that because bacteria like their own kind of thing. But yeah. Sorry for like the really convoluted answer. It's just really complicated. And there's like so many different DNA sequences that are non-coding that can interact with RNA polymerase. But it's the promoter sequence and like the fact that RNA polymerase recognizes the promoter sequence.
Oh, so you're saying that it just like, okay, I got it, I think. Yeah, thanks.
Yeah, you just need RNA polymerase and then the other transcription factors, which are other proteins, to bind to the promoter sequence and initiate transcription. So that's why it's not all willy-nilly. And when the cell doesn't need transcription to be happening, it can modify those DNA bases to where the transcription factors can't bind and recruit RNA polymerase, so it just blocks transcription from there. But yeah, that's like something I could talk about for like, I don't know, 3 weeks. It's a lot of like specific stuff, and the IB only wants you to know like really basic stuff about it. So yeah, good question though. So ahead of this, like ahead of the transcriptional complex, free ribonucleotide triphosphates are cleaved just like we talked about before, but it's instead of ribonucleotides instead of deoxyribonucleotides. Just a small difference. Those are cleaved, and then the energy from the phosphates being cleaved allows them to join in on the growing RNA sequence. So it's just this long elongation, and then it doesn't look like this exactly. This is just like a simplified model because you don't want the mRNA to really be binding to the DNA strand. But yeah, so your antisense strand is going to be your template strand. Your sense strand is known as your sense strand is going to be like the strands, the coding strand basically, because the mRNA is going to be almost exactly the same in sequence as the sense strand, with the exception of the thymine and uracil difference in ribose and deoxyribose. And after it's done transcribing and it hits like a stop sequence, the RNA polymerase dissociates, and then other proteins and stuff come in and reform the helix. You don't have to memorize the codon table. I don't know if you have to memorize the start and stop codon. The start codon is just AUG, and these are the three main stop codons. My molecular 2 biology professor told me, remember the stop codons as you are annoying, you are gone, you go away, which is like a helpful way to memorize it if you need to. So now we've had this mRNA here, and this mRNA has triplets called codons, so it's just 3 bases at a time, and that's how it's read. So the reading frame is 3 little each in 1 codon. So each codon is associated with 1 amino acid each, and each amino acid may have different codons that code for it. I think this is like a really cool thing because it allows for a lot of protein, like a lot of genetic variety, and also I don't think you have to know this, but it just helps suppressing mutations. Like if you have like a mutation, if you only had like a mutation, if you have like a mutation at AAU, but like you mutate to AUC, it's still the same thing, so you're cool. Okay, so the reading frame starts with the stop codon here from AUG, which codes for methionine. So methionine is going to be the first amino acid, and it stops at the stop codon. The stop codon doesn't code for anything. You might hear the stop codon be referred to as the nonsense codon. I think you have to know about nonsense mutations, which is why like nonsense mutations are called nonsense mutations, because they just like insert a stop codon into like the mRNA sequence just like prematurely. This so this genetic code is almost universal. There's some exceptions with like some bacteria and some archaea that have like really weird nitrogenous nucleotides and nucleotide bases, and they have some modifications. But for the most part, this is going to be like your main almost universal genetic code. And because the genetic code is so universal, that allows you to insert the genetic information from one organism into another organism through a type of gene transfer. So for the genetic code, you're gonna have to be able to like deduce the DNA base sequence from an mRNA. So like if I have an mRNA, you'll be given like an mRNA, and you have to be able to like go back to the sense strand or the antisense strand based on the mRNA you're given, and you also have to like deduce the amino acid sequence from the mRNA. They'll give you the table, so you don't have to memorize this, and so you can be able to like go from the codon to the protein. So the application I mentioned about transferring genetic information from one organism to another is what we use really with transgenic organisms. One example of a transgenic organism is bacteria. Like there's different types of transgenic bacteria we have. One of the more common ones are these bacteria called overexpression. They have overexpression vectors. So they will take DNA or some RNA source to get the gene of interest here, and then the gene of interest, which is like this little red piece, and then they like put it into plasmid DNA, and then you'll get a recombinant bacteria. So like the bacterial chromosome is separate from the plasmid. The plasmid can't associate with the chromosome. It's just like a little piece of extra DNA. The bacteria have like little plasmids that they store like extra genes that they don't necessarily need but could be helpful in, like antibiotic resistance. And the plasmid vector is like really cool for us because that's how we can insert the instructions for them to make insulin. So we splice the DNA into a plasmid vector, and then we give the plasmid vector to the bacterial cells and hope they take it in. And then we use some type of antibiotic or some type of selective method to only select for the bacteria that we know for sure have taken up the gene of interest. And then you spin down the bacteria, you kill them, and then you spin them down in like this like centrifuge, and then from there you can extract the different proteins you want. In this case, it would be insulin. So moving on to translation. So we talked about transcription. So in eukaryotes, transcription is going to be in the nucleus, and then you have translation out in the cytoplasm. Bacteria don't really have bacteria don't have a true nucleus, so translation and transcription are just going to be like at the same time for them. But for us, once we've finished transcribing, the mRNA has to be processed and it has to be sent out through the nuclear pore into the cytoplasm. And then tRNAs are an important part of this. tRNAs have to be charged with the amino acid that they are associated with. So that's a whole process in itself. I think you have to know about the charging process, but I'm not 100% sure. I just like typically do topic 2, topic 8, topic 10 review. It's been a while since I've looked at the other stuff that IB students have to know. But yeah, so your first nucleotide in the ribosome is going to be, not the nucleotide, the first amino acid is going to be methionine. you can see here like the ribosome is made up of 2 subunits. In eukaryotes, it's the 80S subunit, and in prokaryotes, it's the 70S. The 80S is made up of the 40, which is the small one, and the 60S. It's really annoying that 40 and 60 don't add up to 80, but like it makes sense to me in terms of the reason why. I don't think you need to know the reason why. I think you do need to know about the different ribosomal subunits. So bacteria have 70S, which is made up of 30 and 50, and then eukaryotes have 80S, which is 60 and 40. So the process is here. So the anticodon strands, the anticodon, these anticodons here are going to be complementary to the codons. And so each tRNA will have like a specific sequence that it can match up to. And the associated amino acid can, the one in the P transfers to the one in the E, the A site. So you have the ribosome has like 3 different portions. It has the P here, which is the peptidyl, which makes sense. This is the like place where transpeptidation happens. So P here, A here, and E for exit here. So exit site is here, A is where it comes in from here, and P is in the middle where the transfer happens. So like the polypeptide transfers to here, and then it moves over one, and this leaves, and then the whole process starts again. And then it keeps doing that until it reaches a nonsense codon, and then the ribosome dissociates, and the protein is complete, and then you have folding and stuff. I think that's the end of the slideshow. So we can like do practice problem questions if you guys want. Would you guys be up to doing practice problem questions? Just like type it in the chat or something.